Hi, I'm Pat Basu, President and CEO of Cancer Treatment Centers of America and the host of Focus on Cancer, a regular webcast that answers everyday questions cancer patients have about the treatment and delivery of their care, answered by experts. I am particularly delighted to welcome a colleague and friend onto the show today who is an absolute pioneer and one of the most premier oncologists in the world. Somebody who has advanced the knowledge, the treatments, the care in cancer at every corner. Dr. Markman, Dr. Maury Markman is the President of Medicine Science at CTCA, and he's joining us from the hospital in Philadelphia. Welcome to the show, Dr. Markman.
Thank you, Pat. I'm delighted to be here, and I look forward to our conversation about this incredibly important topic of pharmacogenetics.
So today's show is one that I'm really looking forward to. It's about pharmacogenomics, a very exciting area in medicine with incredible opportunities in the future. But before we dive into it, Dr. Markman, just tell us a bit about your background. What were you doing before you came to CTCA?
Before I joined CTCA, which has now been about 10 years, been a wonderful experience. I was the Vice President for Clinical Research at the MD Anderson Cancer Center. Prior to that, I was the Director of the Cancer Center at the Cleveland Clinic in Cleveland, Ohio. Prior to that, I was the Vice Chair for the Department of Medicine at Memorial Sloan Kettering Cancer Center in New York City. Started my academic career at the University of California, San Diego, and did my oncology training at the National Cancer Institute and Johns Hopkins Oncology Center.
Well, incredible background. You've made a difference at so many different places in your career. You're making a huge difference in what you do now. What drives you? What made you go into oncology to care for cancer patients in the first place?
I did I went to medical school at New York University and did my general internal medicine training at Bellevue Hospital, one of the premier hospitals in America, incredible public institution. And I also had the opportunity to participate in the private practice setting at New York University Hospital at the time. The reason I say that is that at that point, it became very clear to me that there was no other area of medicine, and I was in internal medicine, that was more complex than the care of the cancer patient within the realm of medicine, nor was there any part of medicine that was, in my perspective, more satisfying. The patients, their families were so grateful for whatever we could do in the cancer space. And I even saw that as a general internist. And of course, that was decades ago. And I said, both the complexity to be able to have an impact and the feeling of just really doing something for a patient and his family. There was no other area of medicine that was more interesting, more exciting. I felt professionally gratifying than that, even personally gratifying. And I have not looked back for one moment to think I made a mistake. This has been an incredible journey, both at the scientific and at the personal level, dealing with cancer and cancer patients.
Well, we certainly don't think you made a mistake either. You've had just an incredible impact on the field of oncology and the battle of cancer care. I've spoken to your patients. I've referred some patients to you. You are truly amazing at both the human and compassionate side, as well as the scientific and clinical side. So, again, very delighted to have you on the show, Dr. Markman. Excited to get into really the focus of today's episode, which is pharmacogenomics. A big word, but an important area of frankly not just cancer care, but medicine in general. And so we'll kind of break this down a little bit, but just to get us started from a high level, 50,000 foot point of view, what is pharmacogenomics?
Well, pharmacogenomics is a really important, incredibly complex area. The brief description, which makes it sound not so complicated, though, is simply what is the impact of our genes on the drugs that we take during our lifetime. That obviously goes from the pediatric area all the way to the geriatric area, and that's it. It's a question of what impact does our genes have on those drugs.
That's terrific, and one of the goals, of course, of the show is to take important but complex topics and really break them down into those kind of simple components. So I appreciate you doing that. So let's break it down even further. I mean, you look at the word pharmacogenomics, and pharmaco, meaning the drugs and the effect that those have on the body, and we'll get to that in a moment. And then genomics, of course, being the critical foundation of who we are in terms of genes and the genome. So why don't we just start right there? What is a gene, and what is a gene's role in our health and on cancer?
Well, Pat, you've just asked some really incredibly important questions. I will start sort of almost at the opposite end of the spectrum, but specifically because what I'm going to mention now is what we're not going to talk about later, but it's really important, at least in this conversation. But it is really important in cancer and cancer management, and that is the genes within the cancer itself. And that's a very important conversation. These genes drive the cancers, they drive their growth, they drive the resistance to therapies. So I'm going to put that aside. So it's, again, the specific parts of the DNA of cancer cells that relate to that cancer. So turning to the much broader topic of what are genes within all of us. Genes are really described as the basic functional unit that determine our hereditary and what it is that makes us who we are. It's estimated that there are somewhere between 25,000 of these units within our DNA, and they control proteins that we make that control normal functions, but they also control regulatory factors that determine how proteins work. They are very important. They, again, as I said, they're the foundation of the biology of what makes a living organism a living organism.
I very much agree with you. I often tell patients that genes are sort of the foundation or the blueprint, if you will, that we sort of the set of instructions that tells your body what to produce in terms of those proteins you described, and kind of what to do next, and so I think that's a great description of how you started. So what specifically then in the role of cancer? So if a gene is a blueprint of our DNA and essentially it's telling our cells what to do next, what is the role of our genes in cancer? Does it predispose one to developing cancer? Can you talk about that a little bit more?
Clearly at CTCA, as you know, Pat, as a leader of CTCA, that the genes and their potential role impact the cancer patient's journey with us. So there are three basic strategies or approaches, you might say, in thinking about how these normal genes, again, these are genes that are with us from the time of conception to death. When I say normal genes, I mean something has happened to these normal genes. They're in our germline, and they can influence that cancer journey. So the first is being able to recognize that an abnormality is present before someone develops cancer, and that might be have an impact on the recommendations of members of the family for additional screening that they should have, for example, in breast cancer, additional more frequent screening or different kinds of screening. Or in colon cancer, if there's a risk because of a genetic abnormality, again, emphasizing they don't have the cancer yet, but they may be a recommendation for more frequent colonoscopies, for example. So the one is simply to know there's that risk, and then to potentially screen for that risk in someone who has the abnormality, the mutation. The second area to emphasize is the potential to do something about this beyond screening. And increasingly, there's evidence that there are certain prophylactic procedures that can be undertaken, generally surgical, but there are also potential medications that can be given to reduce risk. But for example, in breast cancer, ovarian cancer, with the presence of a gene that many the listeners and viewers will know as BRCA, the presence of that mutation may lead to the recommendation in certain individuals for removal of the breast, removal of the ovaries. Again, very carefully considered, because there's data that demonstrates that that can substantially reduce the risk of those cancers and basically improve survival of those individuals. This is, again, prevention of cancer. And the third reason why this information can be very important, and this is relatively new strategy, is that those mutations within the germline have now been able to be shown to be relevant for certain medications one can give to treat the cancer. Because again, emphasizing when I mentioned germline, if it's in the germline, it's also in the cancer. But the presence of those mutations, those abnormalities, can in fact be targeted with specific medications, and that includes now in cancers of the ovary, cancers of the breast, prostate cancer, pancreas cancer, for example, to very effectively treat those malignancies. And these are strategies that are increasingly being used, and certainly being used by our colleagues at CTCA.
That makes a lot of sense. So for our listeners and our audience, basically, the first area is to help predict the future risk that one might have based on these blueprints that one might have to develop cancer. The second would be to inform whether there's some action preventative that should be taken based on that. And then the third that you mentioned there would be specifically to look for ways to target the therapy within a given cancer. Now, you mentioned something that is an important term that you've referred to a couple times, the germline. Can you, for our audience, just kind of define what you mean by that germline, and then talk about the difference between genetic mutations, and other germlines?
Germline is a very important term, and really what it means is what is in us from the moment of conception in the DNA. And as we all know, when the egg and the sperm come together, half of our inheritance comes from the male and half comes from the female. And that's really the DNA that comes from half of the male, half of the female. Then from that moment of the mixing together of the DNA, from the egg and the sperm, through to the end of life, that's the germline. Now, it turns out that in some of us, and it's various numbers and it depends on the particular condition we're talking about, there are major abnormalities within that DNA that we call mutations that lead to the potential risk for medical conditions occurring later in life. And this is not exclusive to cancer, but of course, we're talking about cancer here. And for example, the BRCA mutation, which we talked about briefly, that can lead to a risk of breast cancer, ovarian cancer, pancreas cancer, prostate cancer much later in life. And those are mutations. Those are major abnormalities within that germline. There are other differences between individuals, which are called variants. They're not mutations. And there are actually millions of these variations that can occur from one individual to the next within the germline. Let me emphasize, these are not mutations. They don't cause major problems, but that is in fact the differences that occur in all of us that make it why some of us have lighter color hair than others. They impact enormous number of things within our lives, within our normal biology and physiology that are small and subtle. And there are millions of these that occur within us, that occur within the germline.
So Dr. Markman, I sometimes tell patients, I use the analogy of, I mentioned before, of genes being the plans or the blueprints. And I sometimes use the analogy of a house. So sometimes I say that a variant is your house is very different than my house. The staircase is over here. The bedroom's on the right side versus the left side. Those are normal variants. Whereas a mutation or something that could put us at risk might be something where something isn't up to code, right? Or something might predispose to a risk. So a risk of fire if your oven, your stove is not appropriately connected or is in the wrong location. So I sometimes use that as kind of as an example, and that is not a guarantee that there's going to be a fire, but it is a increased risk due to something being out of code and that being a genetic mutation. The last thing that I think I just wanted to sort of clarify for our audience is in the popular science fiction world, I think sometimes people think of mutations as maybe something that can happen to you, and it can later in life due to some sort of exposure. But I just want to be clear, what you're referring to here is an inherited mutation in that germline that presents a greater risk in future life, as opposed to something, patients have asked me before, okay, if I'm exposed to a chemical or if I smoke, does that cause a mutation in the cell later? Can you maybe just help really just clarify for our audience kind of those two differences?
Yes, I think, again, emphasizing the complexity of the discussion, genes can, within a cancer, obviously cells become abnormal at some point. That's, of course, what happens with cancer. So there will be normal genes still within the cancer, of course, but there are some genes that will become abnormal later, and they become unique to that cancer. So we call them tumor-associated genes. But what I'm referring to are the genes that are present at the baseline within the germline that will always be with us. So if there's mutation at birth, or again, before birth, in this gene that has a susceptibility to cancer, that will be with us throughout. Can there be mutations that occur later in genes because of exposure to, as you noted, sun or carcinogens? Absolutely.
That's terrific. Yes, I think it's really important to clarify for our audience, because I've definitely had patients who sort of think about those two terms. One, whether it's the sun is something that happens later and we should think of that as an alteration that occurs later due to some sort of action or exposure. What Dr. Markman is focused on in the prior description is really something that you begin with, which is an intrinsic, like I said before, the blueprint presents a likelihood, not a guarantee, but a likelihood that a patient might develop cancer later. So I think it was really important for us to lay that foundation in terms of genes. I mentioned before at the top of the show, the topic is pharmacogenomics. Let's just briefly talk about the pharmaco part of that before we dive deeper into pharmacogenomics. What is pharmacology, generally speaking? Just kind of lay that foundation for our listeners so that when we get into pharmacogenomics, we understand genes and we understand pharmacology, and we can kind of put them together.
Yes, this is now we're switching topics to come back to the genes. So what is pharmacology? It is really the study of what happens to a drug, any kind of drug. This is obviously a foreign substance that we take. Now, obviously, it has a purpose, this foreign substance, to have a favorable impact on some bodily function. And again, I'm talking in general terms because pharmacology is a very general science. So you take a drug for the purpose of having a positive effect. And the study of where that drug goes, the impact of the drug on the body, the concentration of the drug that needs to be present for a positive effect, the concentration of the drug that will produce a negative effect, all of that is under the science of pharmacology.
That's right. And if we just take a very basic example, Tylenol, the average dose for most people might be, let's say, 200 milligrams of Tylenol. But if you really think about it, its effect in your body differs from you to me to somebody else based on a number of things. Our weight, our metabolism, the amount of muscle mass, how our liver processes that Tylenol, how our kidneys are excreting it from our bodies. And for any given person, that can lead to different doses or bioavailability within the body for varying amounts of time. And there's a concept that we sometimes refer to, Dr. Markman, that you often refer to of the therapeutic window. The idea that there is a window, or I sometimes call it a Goldilocks zone, where in order to get the intended effect of the drug, if you have too little, then you're outside of that window and you're not going to achieve what you're looking to do. And if you have too much, you're going to do damage. So people who have taken an antibiotic before, if the dose isn't high enough, you're not going to kill the bacteria that's causing the infection. Of course, if the dose is too high, you can have toxicity and other side effects. And so basically, Dr. Markman, this idea of a therapeutic window and pharmacology, I think a couple of concepts. First is that, for our audience, each of us has a different therapeutic window for a given amount of drugs, but there are some drugs that have a wide enough window that, given those differences, the same dose can be effective. But there are some where it might be a narrower therapeutic window where a given dose for you and a given dose for me might actually not have the same intended effect. Is that right?
That's exactly right. And again, it sort of emphasizes the science and to some extent the art of pharmacology, because we are, making reasonably educated guesses for a population of individuals who are receiving a drug. And you gave a wonderful example of Tylenol. I mean, there probably aren't too many people in the United States who haven't taken a Tylenol or the generic version of it. And the vast majority of individuals tolerate it very well. And the reason is because as you noted, Pat, there's a very large therapeutic window, meaning that where effect is seen versus toxicity, it's a large range. But that isn't necessarily true for all drugs. And particularly when we talk about anti-cancer drugs, which, as all know, are potentially very toxic because by definition, the goal is to kill cells, of course, cancer cells. That therapeutic window can be quite narrow. And so that's where we get into those concerns.
That's right. And cancer drugs are absolutely the best example of a much narrower therapeutic window in general. That difference, that margin for error is much thinner in terms of too much being toxic, not enough, not killing the cancer. And so that's where we have to be much more precise. And indeed, this is where pharmacogenomics really comes in, right? Because those underlying blueprints predispose, already there's a certain narrow therapeutic window, but on top of that, the effects of your genes and my genes just on the variants, without even specifically talking about specific mutations, just generally speaking, can really make an impact on the way that one's body is responding to medications. So maybe Dr. Markman, if you agree with that, give us an example.
Yeah, Pat, I think that you've laid it out very clearly, and because I was even thinking as you're describing the in your own field of radiology, the importance you give a lot of drugs to give medications to patients prior to, during the procedure, and you have to be very aware, of course, of the potential impact of that and the patients that are receiving these procedures. Clearly, we've talked about the variation from patient to patient. So the idea of the pharmacogenetics concept is, wouldn't it be wonderful if we could potentially predict prior to giving a drug that this individual may have, let's say, more toxicity because they metabolize the drug slower than another individual. Conversely, we might say that this individual may not have as good a therapeutic effect of a drug because this is a drug that needs to be activated when it's given to a patient. It's not active in its normal form. In other words, it's given to the drug, gets metabolized to become active. But if we know that a patient has a particular variant that says, gee, there'll be a slower amount of activation, then the drug, maybe we need to give more of it. Wouldn't it be wonderful if we could predict that, knowing before we even give the therapy through this concept of pharmacogenomics? Remember, this is germline. It's present from the time of conception. So you could have this information, this blueprint for each individual patient, in theory, of course, and then use that information. So there have been lots of examples in oncology where this concept has been explored. And perhaps I might say the example that is most discussed within the oncology community is that of the drug tamoxifen. Tamoxifen is a very widely used anti-cancer agent. It's an oral drug for the treatment of breast cancer. And it has been known for well over a decade that this drug is actually, when it's taken, it is not an active anti-cancer agent. In fact, it needs to be activated in the body to become active. And it turns out that there are variants of the genes that are responsible for this metabolism that do it particularly slowly. And as a result, there's some evidence that suggests that patients who are slower metabolizers, in fact, do not have as favorable effect on their cancer when they take this drug. And that has been ongoing research now for many years looking at this particular question.
It's really very, very fascinating, and I think our audience can see the power of this, which is the idea that we can predict in advance whether a certain drug, and frankly to what extent, might impact them differently and perhaps actually be life-saving, certainly in the cancer arena, is really powerful. It's tying not just, as we said before, the pharmacology, but the underlying premise of those genes, of those blueprints, to predict which drugs will work, to what extent, and the dosing, and where are we kind of in the advancement of pharmacogenomics, Dr. Markman? Maybe just give us sort of a timeline, or are we kind of at the cusp of being able to really predict this for a vast majority of patients and a vast majority of drugs? Or are we more at the beginning, kind of in the sort of theoretical phase? Give us a sense of sort of where we are and how applicable this is today.
Well, I think first of all, as you can obviously tell, I'm a proponent of this approach, and as I emphasized earlier, and I'll say again now because it is really relevant, we are talking about cancer, but this concept is applicable for any medication that any individual takes potentially for their lifetime. So, and it could relate to cardiac medications. It could take medications that people are taking for anxiety or for antibiotics, as you mentioned earlier. And so cancer is a component of this, a critically important component, but still a component. So the direct answer to your question is there ongoing research by investigators, quite frankly, all over the world in large consortia that are looking at very large databases of individuals who are getting medications for a variety of reasons, also studying the variants that they may have. And many of these variants are 1% of the population or 2% of the population. So you need lots of experience to be able to see if a particular variant, which may be hypothesized to impact an effect on a drug, but you need to follow this, and you need to follow large numbers of individuals to see if, in fact, that hypothesis is correct. So my prediction is over the next 5 years, maybe a little sooner, maybe a little later, we're going to see this concept widely used throughout medicine, including in the cancer space, routinely using this information to potentially determine whether a particular drug or a regimen, which might be a combination of drugs, is more likely to be beneficial for an individual patient than another group of drugs, based upon this information that is present, again, in all of us as variants in our germline.
It's incredibly exciting, and I definitely share your passion for this. It's really remarkable the speed and the dynamism at which this field, genetics in general, genomics, but pharmacogenomics in particular, is moving. For our audience, there's a concept some might be familiar with, something called Moore's law. It relates to the computers and the processing speed, essentially. How fast that processing speed can double, and it doubles on this incredibly fast exponential rate. If you look at how fast we have moved in medicine in terms of unlocking our understanding of the human genome and being able to then act on it, as Dr. Markman is talking about, it's really remarkable. I mean, to give you some context, in 1990, it took 13 years and $31 billion to fully sequence a gene. Now we can do that for a couple $100, and it takes about a day. So the progress we've made is just rapid and incredibly dynamic. So with that, Dr. Markman, maybe paint a vision for us. 10 years from now, what might a cancer patient potentially be able to expect? Could they, or maybe even a patient without cancer, could they go in and have their gene sequenced and then be told that this given drug at this given dose is much more likely to potentially save your life than another one? Maybe kind of just paint the potential for this down the road.
Absolutely. And before I respond to this question, this is a moment that I obviously need to distinguish again the cancer genome versus the germline. The issue of the cancer genome is that it changes so rapidly. There's so many differences with individual cancers. And a patient with a cancer may have their genome that changes. So that is a very complex and a very different discussion to have. But back to the question you've asked, the germline. I absolutely see a time, and not that distant of the future, where each individual will have that information of their germline, the various variants. It'll be theirs. It'll be completely secure. It'll be private, whether it's in the cloud or it's on a pin drive or something new that we'll have in the future. That information will define partially any medication we receive. I can see that patient going into an emergency room for some condition, and immediately, before any medication is given, the information regarding those variants will be known to the emergency room doctor, even though that emergency room doctor will have never seen that patient before. And that information will help determine particular drugs that a patient perhaps should receive in preference to another, or medications they should not receive at all. That will become routine. It'll be common. It'll be in the pediatric, in the adult, the geriatric settings. That in your area of, I can see a patient, going in to get a procedure, and there are a variety of medications that the radiologist may want to give. They'll use that information to know what are the drugs we should give or perhaps not give. You'll have that available at your fingertips as well to help that patient. Knowledge of that variation will be very important in radiology, for example.
Remarkably exciting. And again, thank you for making that distinction between that patient blueprint, their germline, what is with them, versus things that could be changing rapidly in a given cancer. So exciting future. What are some of the challenges? What's kind of holding us back right now from making that future state the present?
I think we sort of alluded to it, but I'll say it directly. It's each individual event that we're potentially talking about, that individual patient getting that individual drug, is relatively uncommon. A member of society walking around is not going to know that they might need this particular drug at this point in time. Why would one care about that, or that patient care about it? It may be 1 out of 100 individuals who may be affected by a particular drug. However, when you take all of the possible variants that, again, are fixed from conception, and you take all of the possible drugs that an individual may have in their lifetime, and you have that information in a single database that is secure for that individual, then the opportunity for that individual benefiting from this information at some point in their lifetime when you add the hundreds of different drugs that individuals may take, maybe thousands, you develop a concept where one can say within a health system, you know what? This information is very valuable. We ought to get it. We ought to obtain it. Somebody ought to pay for it. We ought to figure out a place where we can keep it securely for that individual. So it's not that single patient, rare mutation, a drug you'll never know if the patient will ever get. It's the entire universe of drugs that a patient may receive during their lifetime, potentially. The potential variants that may impact lots of different drugs, not just a single drug, that will make this both cost effective for society, but also cost effective for individual insurance plans or employers who will encourage this. Again, the key is this will always be secure. The only person that is relevant to have this for is for that individual patient. That's where the information will be valuable. So I think we will shift from this idea of this individual rare event for that individual patient to a composite. This is valuable information across a lifetime for individuals, and will have a major positive impact on society, costs of society for adverse events, for example, improving outcomes. That will make this an imperative as part of our healthcare system.
It's a great point. It's a great way of framing it. And along those lines, Dr. Markman, this is such an exciting field scientifically and the progress, but one of the challenges and the arts of medicine is, of course, how things translate to communication, how things translate to decision making, and in particular, shared decision making. Involving the patient in those. This is an area where everything from obstetrics, with genetics around, patient risks, to obviously where we're focused on, in talking about cancer. These are complex subjects with a fair amount of sort of statistical analysis that not only does the doctor need to have a grasp of, but then needs to be able to explain that to the patient in a way that they can come to sort of a shared decision making in all of these fields, right? From obstetrics to cancer. So I just love to get your thoughts on, how are we changing and evolving as a society in translating these complex studies and statistics to the bedside, and then in communicating it to the patient to really drive effective change and greater safety and greater efficacy of drugs.
Pat, you've emphasized a really critically important point. Our knowledge may be escalating at a revolutionary speeds. You mentioned Moore's law, but our ability to communicate complex ideas has not changed much. Humans are humans. They have their hopes, they have their fears. They have individual needs in terms of how that information needs to be provided to them. The fact that we have this massive amount of information doesn't change the complexities of communication. One of the terms that has been used that I like, because I think it emphasizes the point, is we need, that is, we being the physician community, and obviously we're talking here specifically about oncologists, but you can generalize that to all physicians and patients and their families. We need to come up with better strategies for decision support, because ultimately, as you highlighted, it is a decision of a patient, generally with her or his advisor, their family, and of course that's going to include the physician. But we need to figure out ways of supporting that decision, whether that is in terms of simplification, not oversimplification, because you can make something so simple that you're not providing adequate information to make the decision, but tools to do this. Obviously, we've talked a lot about decision support and artificial intelligence strategies to help. make things that are very complex simpler. They'll never be simple, but I'm hoping in the future we will come up with strategies that can take these very complex concepts, this very complex information, and provide it in a way that does allow that patient with, again, her or his advisors, after asking questions that are essential, to be able to make those informed decisions.
Yeah, I agree with you. You and I get a lot of questions even from physicians asking about, what is maybe, if we go back to that FU example, what is the risk in a patient who has this, this germline mutation, let alone, let's say that risk is 1 in 10. Okay, well, what does that mean in the context of the patient's therapy? 1 in 10 to one patient might mean something that they don't want to do or that we shouldn't recommend doing versus to another patient that we should. And sort of the balance of kind of risk benefit analysis and the context of these complex calculations and statistics, as well as just communicating, I think is of equal importance as is the advancement of science. And you and I both spend a lot of time on that communication and that edification. And I agree with you. I think technology certainly has a tremendous role to play in making that more accurate, but also good communication with our patients does as well, which is a good segue kind of into the final part here, Dr. Markman. We've got a lot of questions from patients who email us asking us questions and would love to just share a few of them with you and get your responses. On today's topic, this patient asked, aside from studying how we might respond to existing drugs, are scientists far enough along that they're using pharmacogenomics to develop new drugs? What would you say, Dr. Martin?
Yeah, I would say that this is a topic for the future. We are increasingly, when I say we, it's this large we, because I am personally not a drug developer, but the individuals who are really focusing on drug development are increasingly using unbelievably sophisticated models that, again, going back to this area of artificial intelligence, knowing the structure of drugs, knowing the structure of the cancer, knowing the molecular abnormalities down to just unbelievable detail. Yes, the potential would certainly exist to develop drugs, to tweak drugs, to use this kind of incredible design strategy to impact in a positive way potential issues of the variants that are present within some individuals in the pharmacogenomic area. So yes, that is a topic for the future. It is not today what is being done, but absolutely I could see this for something happening for the future.
Absolutely agree. Here's another one. This one is from a patient, Dr. Markman, about the power of knowing your hereditary cancer risk. We have a lot of colorectal cancer in our family, and I've been doing some research, and I wonder if I have Lynch syndrome. How may I find out?
Very good question. This is an area that is, of course, precancerous until there is a cancer. I would certainly suggest a consultation with gastroenterologists. There are genetic tests that can be done, and that will. These are defective in our germline, and that information can be determined with basically genetic tests.
Thank you. And another one somewhat similar from a patient who said, my mother died of breast cancer. Very, very sorry to hear that from this member of the audience. But question is, my mother died of breast cancer. Does that mean that I will inherit the cancer from her?
A very common question. The The fact is, and these are facts. This is not something that I wish would happen. I wish, in fact, the exact opposite. 1 out of 8 women in this country will develop breast cancer during their lifetime. Now, that doesn't mean in any way, shape, or form that 1 out of 8 women will die of breast cancer, will develop breast cancer. The reason I say that is because breast cancer is common, very common in women during a lifetime. And so to say that, again, this individual is asking about their mother, the fact their mother had breast cancer, that doesn't mean that they will have breast cancer. Obviously, there's that potential, but it simply because it turns out breast cancer is a cancer of aging, like many other cancers are. That is certainly not a given that individual will have a family history. Now, the specifics of that, of course, one would suggest that this individual was asking the question, talk to their own oncologist, potentially their mother's oncologist, because they didn't, or their primary care physician. And, a detailed history might be taken. There might be other members of the family who have had cancers that would raise the concern about a potential hereditary risk. But again, direct answer to the question of the fact that this individual's mother had breast cancer does not mean that they, in fact, will inherit or have inherited a gene, or that there is a gene in that particular family that we can identify that increase the risk of breast cancer.
Thank you. And then a final question. Obviously, we as a nation, as a society, still continue to battle this raging COVID-19 pandemic. Clearly, continues to affect everyone's lives and is on everyone's minds. So, one of the questions here, very specific about COVID-19, with current or upcoming COVID-19 trial vaccines in the future, how should cancer patients approach decisions about which vaccine would be safe?
Wow, what a question. I think that we're going to get a lot of information over the next several months that is certainly not available today to answer this question. It's absolutely extraordinary as someone who's been involved in clinical investigative medicine now for almost 4 decades. The speed at which the pharmaceutical community, investigators around the world that are working on developing effective vaccines is truly extraordinary. There are, at least a half dozen, if not a dozen potential candidates for vaccines that may, quite frankly, revolutionize our view of the management of this horrible virus. All of that being said, we simply don't know today any information that can tell us either the most effective vaccines, the safety profile of the vaccine, which vaccines might be most relevant for a population that, for example, is elderly versus perhaps younger. What about the cancer survivor? Is there, again, this is a rhetorical question because we don't have the answer, a population of individuals who have had cancer, recovered from cancer, might have been treated for cancer with drugs. Is there one vaccine that might be better? That information is critical. Hopefully, and I have no direct knowledge of this, cancer survivors, not necessarily individuals who are receiving anti-cancer therapy, because it is unlikely that they would be included in trials, that is active cancer, individuals that are receiving anti-cancer therapy, but individuals who have a prior history of cancer, hopefully they are being included in some of these trials so we can get some information that's relevant to that population. But the bottom line is, hopefully over the next several months, we'll have a lot of information with a number of vaccines to directly address the critical question that's just been addressed.
Well, thank you. This has been a very enlightening dive into pharmacogenomics and the impact that it has on cancer. I think a really exciting lens into the world of genes and what the potential holds in terms of unlocking the secrets there. We could talk for hours about some other topics related to this for sure. I'd love to just conclude by asking you any advice that you have for patients or final things that you'd like to share for our audience?
I would just say for individuals who have cancer, who are receiving cancer treatment, the advances that are occurring on all fronts, from modern surgical techniques to radiation strategies that improve efficacy and decrease toxicity to, of course, my area of interest in anti-cancer therapies, the changes are just incredible. Advances leading to improvement in the survival and critically important quality of life for cancer and cancer therapy for patients. This rate of improvements are accelerating, and the future holds a great promise for individuals who are diagnosed with cancer. And of course, we want to find much better ways of finding diagnosis, make a diagnosis early, and very importantly, in preventing cancer.
Well, Dr. Markman, thank you again for taking time from your very, very busy schedule to come on the show. Incredibly fascinating topic. Thank you for sharing all of the latest and greatest updates. And I'm really looking forward to doing a deeper dive with you on the next episode where we're talking about advances in precision medicine. So thank you so much for coming on.
Thank you, Pat. I really enjoyed it. Very important topics. The changes here are so exciting for our patients, and I look forward to our next conversation.
Outstanding. Well, thanks again, and we'll see you on the next show.